of pollution incidents. Compliance and enforcement, which is very important, because once granting applications, we need to ensure that applicants are compliant and that we enforce whatever regulation it is that we need to enforce. Spatial and planning, and this is where we get involved in the orderly development of our urban and rural areas and ensure that development is taken in a very orderly manner. Policy and research, this is where we would develop our policies and plans, standards, legislation, which would be relevant to the wider public and the agency carrying out its legal functions. For instance, if it is that you will be conducting your SBA on water quality, our standards on water quality would be a very important resource for you. So you would need to have those standards to judge whether or not the samples that you have collected, if they are up to standards in comparison to the agency standards. Last but not least, public education. And this is where we go out into the communities, we speak to people, we go to various sessions, and exactly what is happening here now, we are making you aware of the resources that are within the agency, as well as we have brought technical officers to speak to you on the various topics within your SBAs. Now, accessing NEPA resources. So we have quite a few resources available for you. So we have resources on climate change, coastal degradation, coral reefs report, which many of you will be, I know will be conducting your SBAs. And so we have documents. The agency conducts monitoring and surveys on coral reefs and the coastline of Jamaica. So those reports are readily available for you. Monitoring reports, water quality reports. Now for the water quality reports, we ask that you ensure that you make your request early, because this may take some time for you to obtain those. Other reports and documents, air quality reports, maps. We have maps which you can obtain, various maps that you can obtain at a cost. Deforestation, parish profiles, and environmental impact assessments. These will be very important when it is that you are writing your introduction for any of your research papers. The environmental impact assessments provide such information as the ecosystem of a specific area, the biodiversity as well. So these are important documents that can assist you. Policies, standards, law, regulations, and guidelines. As mentioned before, the agency prepared these policies, legislations, regulations, and guidelines, and they are available. State of the environment reports, they are more general, but they do provide a wide cross-section of information on various areas. And most of these documents can also be found on our website, as well as the library. We have a newspaper clippings database, and since 2010, we will be able to provide you with any information, major environmental incident or planning incident that has occurred in Jamaica, or any information on the National Environment and Planning Agency. We should be able to provide you with those electronically. Development orders and plans, and this is what the agency uses in ensuring that our spatial planning is carried out with regards to the mandate of the agency, and they provide quite a bit of information. And so another very important document that you have access to and we can provide access to. Now, I spoke briefly on the website, and we have a new and improve website. So we invite you to go there and you will find a wealth of information on our website. Whatever it is that you're unable to get from our website, we invite you to call the agency or the library, and we will be able to provide it for you as well. But go take a look, it's a beautiful site. We also have an e-library where we're able to provide students documents that we have electronically. We're able to provide you with those documents. Once you email us or call us, we can send those documents to you electronically. Very important, other libraries and information points. Now, although the National Environment and Planning Agency is the leading agency with regards to environmental issues within the country, as well as resources, we also need you to know that there are other agencies that may be able to provide you with some of the information that you require. So, for example, specific information on the forestry department, if it is a specific forested area that you are seeking information for, while we'll be able to provide you general information on forestry, we would invite you or refer you to the Forestry Department of Jamaica. Likewise, for underground water resources and the status of those, we would refer you to the Water Resources Authority of Jamaica. Access to information. All right, so this is very important. Now, some of the documents that you may request of us from the library may not be readily available to you. And so what we may ask you to do is to apply under the Access to Information Act. However, unlike the resources in the library that may be readily available and we can forward those to you within, say, 24 hours or dependent on where the location of that information. With access to information, we have up to 30 days to grant that request. And there are quite a few documents that may fall under the access to information, such as water quality reports. So what we ask of you students is to ensure that you apply or make your request on time before 30 days, because we have up to 30 days to provide you with that information. Now, some of the services we offer, referral service, as mentioned, access to the newspaper clippings. We have an electronic database which is also accessible from our website at nepa.gov.jm, and you can search that database and you'll be able to see some of the documents and resources that we have real time. And if it is that that document is not available electronically, we can you may have to come into our offices and view those documents. We also assist with interviews. From time to time, students wish to interview officers. That we ask that you send those requests in quite early, because it would take some time for the technical officers to respond to the interviews. We also ask that whenever you require an interview, that you send us the questions that at the same time that you are making the request. Water quality testing. The agency provides water quality testing to schools, and you can call us and arrangements can be made. So for those who will be, their SBA will be on water quality, yes, we do provide the services to schools. We do test the samples for you, as well as we sometimes go out in the field with the schools, but you'll have to call us for arrangements. I had mentioned maps before, so won't go into that again. So if it is that you need to make contact with us, we invite you. You can walk in, but as you know, with COVID, that may be an issue. So we ask you, instead of walking in, to give us a call and we will make an appointment for you to come in. You can call us, and our numbers are on screen, so you can call us there at those numbers. You may also email us at doccenter@nepa.gov.jm, or you may find us on the World Wide Web, nepa.gov.jm, Sharon O'Connor Documentation Centre. Now, just to provide you with a few tips when you're writing your SBA, although we'll be having a teacher later on to go into that in extent. When you're starting to write your SBAs or any research paper, we ask that you know what is required of you. So speak to your teacher once you have decided on an area that you'll be preparing your SBA. Create an action plan inclusive of timelines, so that will keep you focused. Create a checklist so that as you go through, you can check off what tasks, what activities you have completed. Another thing is to prepare drafts and prepare them in multiples. And when you're preparing your drafts, ensure that they're proofread by someone that will be able to give you reliable feedback. Have backups. This is very important. So at this point in time, we have many things that are on our laptops or flash drives, which can be volatile. And a suggestion or a recommendation is, even while you may download on your devices, on your flash drive, email the resort, your paper to you so that you can have copies, or upload to the cloud if you wish. Cite your resources, and if required, if citation is required, and there are many apps or software that can help you with that. One of the popular ones is All Purdue, and with regards to proofreading, you can use Grammarly. There's a free version, so that will help you in proofreading, help with proofreading your documents. So we will be moving on to the technical presentation. So I do hope you will have a good day, and please remember, if there's any information that you require, you may contact us at any time. Thank you.
Okay, thank you so much, Ms. Miller. And for persons that you may not have gotten the number to contact us, it's 754-7540, or you can contact us by email, pubed@nepa.gov.jm, or doccenter, docentre@nepa.gov.jm. I hope that students, that you would get an appreciation of what sort of resources exist at NEPA and how you can access those as you seek to gather secondary resources as part of your completing your SBAs. So we will now move into one of our technical presentations, and that will be on mangroves. So our next presenter is Delroy Thorney, and he is the outreach officer at the UWI Discovery Bay Marine Lab and Field Station. He is a graduate of the University of the West Indies, Mona, with a degree in the double major environmental biology. Through his job, he reaches the minds of many and informs them about the importance of the environment in which we all live and should cherish. Delroy is also a PADI open water diver with a love for keeping the environment in a healthy manner for future generations. He believes that if we take care of our environment, it will take care of us. So Mr. Thorney, over to you.
All right, good morning, everyone. My name is Delroy Thorney, so I was just introduced by our host, and today I'll be giving you a lecture on mangroves. So I'll just share my screen so you guys can see. I'm joining you all remotely today from the Discovery Bay Marine Lab. All right. So today we'll be talking about mangroves, the role and the relevance in which they play. And this presentation is brought to you from the Discovery Bay Marine Lab, along with our partners from the Living Oceans Foundation, the jamming program in Jamaica. Now, this is the outline which we will follow for the presentation. We have what are mangroves, distribution of mangroves, the types of mangroves that are found here in Jamaica and the Caribbean, ecological importance of mangroves, mangrove habitats, their protection that they offer to us, and threats to mangrove and socio-economic value. So let's jump right into the presentation. What are mangroves? Now, mangroves are classified as trees and shrubs which live in the intertidal coastal area. They're only found in the tropics and the subtropics. So when we're talking about the intertidal region, we're speaking about regions which are influenced by the sea's gradual rise and fall in water level on the coastal area. If there's no fluctuation in water level, then mangroves don't really persist and live as well. And these mangroves are what we call halophytes. Now, halophytes, if you break down the word, halo stands for salt and phyte means plant. Now, these plants are highly adapted to living in salty conditions. And as such, they are one of the few organisms, plants, that you find persisting in the coastal area. So we're talking about where they're found. As I said, they're found in the tropics and the subtropics. Now, that's found here between 30 degrees north and south of the equator, highlighted in green. And we have a few outliers just within that region, just because there's where we have warm ocean water currents carrying water that is tolerable for the mangroves within that region. If it gets too cold, the mangroves will die. So here in Jamaica, we have mangroves all around the coast of the country. Where I am right here, we have mangroves, a small amount here, and we have mangroves. If you look on the map, you notice you see these blue lines connecting to each major mangrove. Now, these blue lines represent rivers, and wherever you have rivers, bring in fresh water, and the mangroves tend to exist in what we call brackish waters. So the waters which they're washed by every day is not only salt water, sometimes there's a flushing system going on. So the fresh water from the rivers kind of flushes the system and maintains a balance. So it's not completely salty. Like since seawater is 35 parts per thousand, sometimes brackish water it gets to 15, 20, 25, and so forth. So they are influenced by fresh water and salt water. They can exist in either one, but they are able to tolerate salt water very well. So the presence of mangroves in nature is dependent on a few factors. These are elevation, tide level, salinity. Hold on, we'll be taking questions at the end of everything. So elevation, tide level, salinity, and wave action. And when we have plants which are existing along the coastal area, these 4 factors are very relevant to their existence. So the elevation along the coastline, the tide level, whether it's in the lower tidal zone right here, or they are in the high tide zone up here, you tend to find that there is a presence of separate species which creates something called zonation. As you can see here, you have red mangroves, we have black mangroves, and we have white mangroves. So if it's in the low tide region, you'll find the red mangroves. As you can see, they have specialized roots right here, which can help them adapt to live in that area. And the plants which are normally within this region tolerate the most salinity. All right. So there are two main groups of mangroves. There are true mangroves, which are plants which exist in completely salt water, and they're also influenced by fresh water, but they have the true adaptations which make them mangroves, which are features such as vivipary, which vivipary just basically means that these plants have their seeds germinating while they're on the plant. So the embryos develop while still attached to the parent, and once they detach, they start rooting and can grow immediately. And then we have the mangrove associates, which are plants which are found in vicinity of the true mangroves, and they also help to create the mangrove forest. And worldwide, there are about 54 true species of mangroves. In Jamaica and the Caribbean, we only have 3 true main species: the red mangrove, Rhizophora mango, the black mangrove, Avicennia germinans, the white mangrove, Laguncularia racemosa, and then we have the button mangrove, which is the Conocarpus erectus. Now, we'll jump right into the red mangrove. Give you guys an idea of what the mangroves are like, and so we can talk a little bit further about them. The red mangrove are trees which have specialized roots called prop roots, and area roots called drop roots. They get their red color from a compound called tannin, which is usually used for dyes in some areas of the world. So if we look at the mangroves here, we can see them spread out along the coastal area, washed along by the seawater. So coming from up here, from the stem, we have what we call drop roots. Now, both drop roots and prop roots have what we call lenticels. These are special breathing pores on the mangrove that allow it to have gaseous exchange. Since the roots are submerged, normally these plants, the roots need to be breathing. So they develop these drop roots to get aeration from the stem and along the stems of the prop roots. Now, the prop roots are designed to withstand wave action, strong wave action from the shoreline. So you find that they are sturdy and they branch out to keep the tree balanced. So in times of storm surges, it kind of breaks the waters coming into the land. And these trees have a special adaptation. They have what we call a sacrificial leaf. These sacrificial leaves absorb most of the salt that is absorbed into the plant and deposit it into a few of the leaves. As you look on the plant, right, you see a lot of yellow leaves. These contain a high amount of salt, and once they reach maximum capacity of salt, then they fall off the tree and then recycle that salt back into the sea or the mangrove muddy waters. So now we're talking about black mangroves. Now, just as the name suggests, they have a black bark. That's how they got their name. And they do not have prop roots like the red mangrove. What they have instead are called pneumatophores. Now, these pneumatophores, they are special adaptations, just like the lenticels on the prop and the drop roots of the red mangrove for breathing. So since they're found further inland, where there's occasional flooding when you have high tide rising, you have these long and pointed roots that point out of the ground to help it to breathe. And better example you can see right here, all these sharp looking objects sticking to the ground, these are the pneumatophores. And just like the red mangrove, they have to deal with salt in a special manner. And as such, you find them excluding the salt through their leaves. So if you look in the mangrove forest, you notice that you see some leaves with some little white patches on them, crystals. These are all mangrove emitting salt from their leaves. So salt is in a liquid form in its body. And then afterwards, it becomes crystals when it is exposed from the leaf and it dries. Next up is white mangroves. Now, the white mangroves, they have a white bark, but if you see them in nature, most times they look a little bit darker due to being sunburnt, but they have a white bark. And they also have breathing roots, just like the black mangroves. They're very similar to them, but they have what we call knob roots. These are shorter roots, which look like these, and they're not as abundant as black mangroves since they are usually found further inland where the high tide does not reach as much. Then you have these shorter roots which are able to get aeration for the roots. And these plants are very special. They have what we call extrafloral nectaries right at the base, right here, the petiole. Now these secrete a nectar which attracts other organisms to create a symbiotic relationship. And this symbiotic relationship benefits both the plant and the organism which it is feeding. For in some instances, ants tend to protect the white mangrove tree from predation from organisms such as snails, which may try to eat it. And in return, they get nectar. And they also excrete salt through their leaves, as you can see right here. All right, moving on. We'll talk about the associate mangrove now, buttonwood. Buttonwood mangrove is a mangrove which is not a true mangrove since it does not have vivipary. It does not have vivipary, and as a result, it is not a true mangrove. So we're to jump into the ecological importance of mangroves right now. Mangroves are major nursery and habitat for a lot of species. As a result, we have a lot of fishes which are commercially important being found there. So a lot of fishes come from the sea, come into the mangrove area, and they spawn and then reproduce so they can have more kids going forward. They also act as biological filters. So this means that they are sinks and pollutants for nutrients. So whatever runs off from the land, it is absorbed by the mangroves and reduce the amount of nutrients going to the sea. And as a result, reducing the effect of eutrophication, which is the presence of a large algal bloom. Then we have carbon sequestration, which basically means it is a major source of capturing atmospheric carbon. So which carbon is one of the main things which contribute to global warming. And when you have carbon sequestration, mangroves are very productive in that manner and take out a lot of carbon out of the atmosphere and store it in their roots, their stems, and in the soil that they exist in. Also, water quality maintenance. A lot of organisms in the sea, such as corals, depend on clean, crystal clear waters to survive and thrive. And mangroves reduce the amount of sediment which goes out into the sea and keeping our seas crystal clear as we see them. As you can see at our beaches, most of them have these clear waters. And as a result of mangroves cutting down the amount of sediments going out into the sea. So as a habitat, many different species are found there. And these are some of the species that we can find here in Jamaica. As you can see, we have things that live in a canopy. We have a lot of birds, pelicans, egrets, frigate birds. And then we have other things that are found within the branches and on the floor of the forest. In Jamaica, we mostly have crabs and snails, but in other countries, such as the Asian countries, you can find tigers. In some countries, you can even find raccoons in their forests and monkeys as well in the branches of these trees. In the prop roots of the red mangroves, you find these organisms growing on them, bryozoans, oysters, and such forth. And then we have things that normally swim through, dolphins on occasion. This picture was taken here in Port Royal, over near Refuge Key. Sorry, not Refuge Key, near the Goodbody Channel, around that side. And protection-wise, mangroves are very important to us. They provide protection against storm surges. When hurricanes come, those red mangroves you see right there, they act as breakwaters and reduce the amount of wave energy coming into land and reduce the amount of flooding. There's also coastline protection from erosion. So where you have lack of hard rock and a lack of plants, you tend to find that coastal erosion occurs very rapidly or quickly. As you guys saw, I'm sure some of you saw Hellshire recently, the coastline eroded very greatly around that side. And then there's also sediment and garbage trap. We spoke about sedimentation, protecting corals from sediments, and it also protects the sea from our humankind's solid waste So plastic, it prevents it from going out into the sea as well. And the accretion of sediment helps to build coastal areas as well. And it also protects us from global warming. So ecologically, it benefits the environment and it protects humanity in general. Now, the major threats to mangroves are man themselves. We tend to drain mangroves for development, dump up the areas so we can make new buildings and for farming, mariculture. In some Asian countries, they wipe out entire areas for mariculture. So they have shrimp farms in the waters around the coastal areas. And this reduces the amount of mangroves that are present, protecting the environment and protecting the humans themselves. And there's also the increase in demand for mangrove wood. Most people tend to burn it for charcoal. So if you go into the forest sometimes, you notice you see some burn spots. These are where people create charcoal kilns so they can create their charcoal to go and sell illegally, of course, because they should get permits to cut down mangroves most times. But these permits are disregarded most times and they just go out and illegally do these things. And then there's also the presence of solid waste. So a lot of garbage plastics are thrown out into the environment and this suffocates and chokes the mangrove. So even though it protects the sea, it's dying in the process. Now for socio-economic value, mangroves are extremely important to the socio-economic status of Jamaica. It is a source of job creation. It can increase the GDP of the country, and this stems from national, foreign, international investments. So this national growth from foreign investments help the country to make more money and be more productive. And it's also a very good educational outreach platform for us. And it also presents us with opportunity for a cleaner environment. And here are some examples of how they are socio-economically beneficial. Glistening waters in Trelawney, these biofluorescent organisms live in the water. These are found near mangrove forests. And the tour, they have hundreds of tours, thousands of tours every year. People want to see this at night, and it brings in a lot of money, foreign investment for the country. We also have Black River Safari Tour, and we have a boardwalk here at Discovery Marine Lab. So educational reasons. So you have ecotourism and you have job creation from these things as well. So you develop these new things in the environment, you have job creation, people getting jobs, more money, people benefit, the country benefits in the long run. And then you have people traveling from the world for bird watching as well. People will travel just to watch birds in the forest, and we have a lot of beautiful organisms that exist in the mangrove forest. And in terms of international investments, we have what we call we have foreign investments. So Spain, in recent times, according to the Observer, they invested US 1.7 billion US dollars to the local tourism sector. And this benefits the country's gross domestic product and helps us to have a better and more productive future as we develop as a nation. And in the process of doing this, they created over 2,000 jobs. This ecologically friendly development, Oyster Bay, Excellence Oyster Bay in Trelawney. This hotel is developed in the mangrove, and it has a nice pristine appearance, which helps people to appreciate nature. And it created over 1,500 construction jobs and 700 staff right there. So over 12 years, UWI has been doing their part to help the environment and replanting a lot of destroyed mangroves, either from hurricanes or from human impact of dumping up on clearing lands. And we do that because mangroves are necessary for a brighter future. And without them, we can't really progress as a nation to become first world status, because the environment has to play a role in becoming a first world nation. We protect the environment protects us, and we both benefit from it. And with that, we come to the end of the presentation. I'd like to ask you guys one question right now to see who has been paying attention. You can raise your hand and accept your response. So the question I'll be asking is, I haven't even asked the question as yet. All right, so what are 3 ecological importances of mangroves? All right, so let's see. So Ajixney Clark raised their hand.
Well, sir, for one, I know that the mangroves can keep out storms and hurricanes. Well, not really keep out, but reduce the impact
Yes,
on the environment, sir.
that's one. 3 ecological importances. Give me 2 more. Ajixney?
Pass, sir, pass.
All right, would anybody else like to attempt that question? 3 ecological importance of mangroves. All right, so Miss Blake. Okay,
Hello, can you hear me, sir?
okay, go ahead.
Okay, good presentation, first of all, sir. Thank you for that.
Thank you.
One, it can be used as a nursery and a habitat for the small fish, little
Yes,
fish. Another one, it filters the silt that is coming
okay, lovely.
river, and
Yes,
and
come on, one more, you're close.
and geez, it helps with the water, like the quality of the water to be.
Yes, that's correct. So Miss Rachel Blake, make note of you so that you can get one of the prizes from the NEPA group.
Yay!
All right, so with that now, are there any questions before we move on? No questions? Okay, so we'll be handing back over to the NEPA staff. I thank you all for being here today. You seem to be very receptive and I'm grateful for that. And I do hope the best for you with your your SBAs, IAs, whatever you may be doing on your exams going forward in the future.
So much, Mr. Thorney, and we will make contact with our winner and she'll be able to collect her prize. So we'll just move right into our next presentation, which is from Mrs. Safia Roden. She is coordinator for the Ambient Water Quality Monitoring Unit within the Pollution Monitoring and Assessment branch of the National Environment and Planning Agency. She has worked at NEPA for the past 5 years, and she knows a thing or two about water quality monitoring and assessment. Mrs. Roden is an environmental biologist who specializes in aquatic resources assessment and is a graduate of the University of the West Indies with a master's in natural resource management. She's an expert at doing things at the last minute and advocates against this practice in the lives of our young people. She is a proud graduate of the St. Louis High School for Girls and aspires to become a world-renowned tea party hostess. Until then, she will continue to help in the protection of Jamaica's natural environment, one sample of water at a time. Ms. Roden.
Good morning, everyone. Thank you all for allowing me this opportunity to speak to you on water quality. You've heard a lot about me. I'm Safia Roden. Once you all get through CSEC and I start having my tea parties, you're all invited. But let's get right into it. Today, we are going to talk about ambient water quality monitoring.
And I will try my very best not to be too long because I don't want to hear any bellies growling over the Zoom. So, all right. So just to give you a little introduction into what it is that we do here at NEPA, we have been conducting routine water quality monitoring since before I was born. And in 2009, we made it a formal program that was developed to assess our coastal water quality, our rivers, to see what it is that's happening with our waterways. The monitoring that we do, it helps us to make decisions at the agency level, and it helps us to ensure that whatever water that we're using for recreational purposes, as well as for the health of our aquatic environment, is of a good quality. So for today, we want to look at what it is that you know, right? It seems my fingers are a little too big for this button. So what it is that you know, right? So these are the questions. This is basically an outline of what we're going to do today. And we want you to just keep these questions in the back of your mind, because you never know at the end what might be happening and what questions we might ask you to look and to say, give us the answer for. So what is monitoring? Why do we monitor? What is pollution? Is water quality really important? How do we determine water quality, and what is a sample? I hope you are listening, because those questions might come back to haunt you later. So what is monitoring? Monitoring is, in simple forms, a process, right? Right. It is a process of sampling. It's a process that involves measuring and recording indicators that will help us to assess conformity to specified objectives, right? And in simple terms, we're looking at what is there and we're comparing it to something that we would want it to be like. We want it to be like a certain standard. We have a standard that we would like the water quality to be at, and so we will monitor, we will sample, we will measure different indices to help us to gather information to see, hey, is this conforming? Is this becoming? Is this being like what it is that we want it to be? Is it conforming to the standards, right? Okay, so why do we monitor? We monitor so that we can know what is happening. In our line of work at NEPA, we are charged with the mandate to manage and protect our land, wood, air, and water. And in order for us to manage it, and in order for us to assess whether there are changes, we have to first know what is happening and what is going on. And so that is why we monitor, so that we can define any status, define the water quality status of the body, to see if there are any trends, any changes over time, and overall to measure the suitability of the water for its intended purpose. And when we gather all that information, we're able to use it to implement management strategies and activities that will help us to fight against the effects of pollution. And that's those are some of the reasons why we monitor. Okay, all right. So what is water quality? So we've been looking at monitoring, and we've been saying, all right, we want to monitor water quality, but what is it? In simplest terms, water quality is the suitability of water for its intended purpose. And we use water for many different reasons. We go to the beach for recreation. We use water for drinking. We use water for planting and farming, and all sorts of reasons and all sorts of intended purposes. And so the standard of quality for water for recreational purposes might be different from the standard of quality for drinking purposes. And so water quality is how suitable the water is for its intended purposes. It is the condition and the environmental health of the water body and the standard of purity that is necessary to ensure that the water body is protected, to ensure that aquatic resources, the organisms living in there are protected and thriving and alive, right? Now, what is pollution? I know you're looking at this screen. You can go back to that screen. I know you're looking at this screen and you can definitely tell that these waters are polluted. We're seeing oil spill. We're seeing dirty water from a drain pipe going into what looks like a sea or a river. We're seeing these two young men having fun in a river of garbage. And you're looking at that and you're obviously noting, hey, this water is polluted. It is nasty. It's disgusting. Something needs to happen right away. But if we go to the next slide, we can ask ourselves the question, is this water as polluted as the next one? Is this water even polluted? Look at it. It's beautiful. You feel like you just chuck off right away in there. Clear, white sun, pretty blue sea. Is it polluted? I want you to think on this. Think on it. Can you see every type of pollution? Can you see every type of contamination? And the answer is no. And so looking at this water body here, we might not be able to tell whether it is clean or polluted. We might not be able to tell whether it is suitable for its intended purpose. It might look pretty, but can you really go in there and take a swim? Can organisms really thrive in there? So let's look at the definition of pollution now. It is the introduction by man, directly or indirectly, of substances or energy to the environment, resulting in deleterious effects, right? And one of the things about pollution is that these substances, many of them occur naturally in the environment. However, it becomes a pollutant when its concentration and the amount of it in the environment is so much that the environment cannot recover from it. And the change is so drastic that the environment and the organisms cannot live and they cannot survive in this environment. And so pollution happens, the water becomes contaminated. Next slide. Thank you. So some of our major water pollutants are microbes or bacteria, nutrients, heavy metals, organic chemicals, oil and sediments, heat, and solid waste. And let's get into just a few of them. So like chemicals, I know some of you might be interested in what the types of water pollution are. Let me repeat that. I know you are very interested in knowing what the types of water pollution are. And there's chemical, suspended matter, microbiological, nutrients, and oxygen depleting. Chemical, suspended matter, microbiological, nutrients, and oxygen depleting. So chemical, these are from metals and solvents that can come from your industrial works, your pesticides, and your petroleum. The suspended matter, you have total suspended solids, and these linger within the water column. And one of the most popular ones that everyone knows about being suspended is microplastics. And these are a growing problem all around the world. So I hope that we're all adhering to the plastic ban and not carrying any scandal bags, right? We have microbiological. These are your bacteria, your viruses from improper sewage, for example. One of the most deadly ones that we can think of is cholera. And you'll find that a lot of the poorer countries who don't have access to clean water, they have persons who are suffering from cholera. You have nutrients that can be found in your wastewater and your fertilizers, and these cause excess algae growth, right? If large concentrations end up in your waterway. And then you have the oxygen depleting. These might be discharged from processing plants and paper mills or wastewater treatment plants. And they're one of the most important types of pollutants because these, as the name suggests, depletes oxygen.
it removes oxygen from the water. So bear those 5 types in mind, right? Let's go on to the next slide. And you have different types of pollutants, different sources of pollutants. You have point sources. These are where you can see a discrete conduit for the pollutant entering the waterway. So you have drains and gullies. You might see oil leaking from a ship, oil spill. Those are your point sources. The next type is our non-point sources. And these are where you can't really tell where exactly the pollutant is coming from. And these occur mainly because of surface runoff from heavy rains, and the water is running over the farm, over the roads, storm water, it's just going everywhere. You can't really tell where it's coming from. So those are your non-point sources of pollution. Next slide. So at NEPA, we monitor several parameters, and how we decide whether the water quality is good. It's not that we're going to go out there and monitor sewage, or we're not going to go out there and test for sewage, nor are we going to go out there and test for fertilizers. But these indicator parameters can tell us and give us an indication that these are the things that contribute to the high levels in the waterway. So our nitrate, phosphate, pH, biochemical oxygen demand, or BOD for short, fecal coliform, and your dissolved solids. Our next slide. So for nitrates and phosphates, those fertilizers that make the plants look pretty and grow and happy at night, they're one of the primary sources of our nitrates and phosphates. They're also found in sewage and wastewater, and maybe not so much now, but phosphates used to be a very big part of soaps. And the effects of them is that they will cause an overgrowth of algae that can lead to blocked sunlight going into the water that can reduce the oxygen levels. In short, that's called eutrophication. Next slide. And we test for nitrates using the cadmium reduction method. And for phosphates, we use the ammonium molybdate and potassium antimonial reaction to test for our phosphates. Next slide. So for biochemical oxygen demand, I said before, you have microbes that will go into the water, and these cause decomposition. And in that process, they actually use up oxygen. So biochemical oxygen demand is a measure of the amount of oxygen that is needed for microbial decomposition. So you can imagine that as they're using up the amount of oxygen, then it's going to be depleted. So when you have a high BOD, you have low dissolved oxygen, and that can induce stress on our organisms and the aquatic environment. And it is found, it can be discharged through sewage and wastewater as well. For our next slide. And we test it by using the BOD day 5 method, where we sample what the dissolved oxygen is on day 1 and what it is on day 5, and we see how much oxygen has been used up in a 5-day period and do our calculations. For fecal coliform bacteria, this bacteria comes from the feces of animal and human waste, and they are an indicator, primary indicator of sewage contamination. With fecal coliform bacteria, it also tells us the presence or the possible presence of other pathogens that can cause gastrointestinal issues for humans as well. And so this is one of our key indicators to see how healthy our water body is, particularly for recreational use. And so we will compare our results to the World Health Organization's recommendation for recreational amounts in the water, amounts suitable for recreational use. This is done using the multiple tube fermentation method. And if you look on the screen, you'll see some of these tubes have bubbles, and that indicates that the bacteria is alive and thriving and multiplying in those waterways. Next slide. Our pH, how acidic and basic a substance is, and most organisms operate at an optimal pH, and so if it changes drastically, it can cause stress on the organisms, and life functions are limited outside of this range. So how do we get all of this information? By collecting samples. And samples are collected in a reliable way. Sampling is the most reliable way to gain information about a whole system. So it is the process of selecting an appropriate portion. So if it is done properly, we can gain information about the whole system, because it is not feasible to sample every stretch of river and all around the coastline of this beautiful island. So as we said, yes, it's an easy and reliable method. And next slide, please. Thank you. So for us to sample, we collect our samples at NEPA in 3 bottles for us to carry out the test that we mentioned before. The bottles on your screen, you have the fecal coliform bottle, which is the one with the foil cap. That is a bottle that is used to test for bacteria. That one is sterilized at high heat and pressure to ensure that it is clean, and whatever goes in is just collecting bacteria from the sample itself. The brown bottle is for your nutrients and your solids, and the rounded bottle is an airtight seal. It provides an airtight seal so that no more oxygen is entered after collection, and this is what we use to test for our dissolved oxygen. And we can test using a boat, we can go out wading out into the river, we collect from the sea, we collect from waters all around in our natural environment. Next slide. So what do we do with this data that we would have, what would we do with this data that we would have collected from these samples? The data can be used, I see someone is making some very fancy notes. The data can be used to, we can go back to that slide, the next slide. Data can be used to show trends in what is happening in the water body and how it changes downstream. So for this slide, for example, this is showing you how BOD concentrations changes along a river from upstream to downstream. For the next slide, we can compare water quality data between years. So for this slide, this is showing you how fecal coliform concentrations changed from upstream to downstream in 2015 versus 2019. For next slide, we can show you a graphical representation of the distribution of the pollutants in the water, which areas have the highest concentrations versus the areas with the lowest concentration. So especially my geography students, get ready, you can use water quality data for mapping in the future. Our next slide. So it's not just a local outlook, there's a global outlook. And one of them, there are many things happening in water quality globally. One of the things is called dead zones. And this is because there's so much pollution in these areas that the oxygen levels have gone down to basically low or zero oxygen. And many organisms cannot thrive and cannot live there anymore. And these are very problematic areas. Looking in the Caribbean concept now, for our next slide. For the Caribbean concept, we have 25% of the regions has access to sewage treatment system, while 52% relies on latrines. And this is problematic because if you have latrines, this means that your pollutants are going down into your groundwater. And if you don't have persons with many proper sewage treatment systems, you will have persons discharging all over, and these can enter your waterways through point and non-point sources. So we want to bear these things in mind as we go along. So these are basically some of the things that we look at for water quality. And I hope you are listening, especially for some of the questions. And so we're going to be asking you 2 questions. And if you have any question, if you have the answer, please raise your hand. And then we will acknowledge you. So the first question is, name 5 types of water pollution. Pardon? Jada? Jada? Go ahead, Jada.
Yes, miss. Are you hearing me?
Yes, go ahead, Jada.
Okay, miss. Chemical is one type. Suspended matters, which include microplastic, microbiological, which include bacteria, viruses from improper sewage, nutrients, which causes excess algae growth, and oxygen depleted.
Wow, Jada, give yourself a hand. You gave me the 5 types and the examples. I am very proud of you. Very proud of you. The second question that we want to ask is define, very easy, define water quality. Casey, go ahead, Casey.
Miss, water quality is the suitability of water for its intended purpose.
Congratulations. Thank you so much for listening, Casey. And right now we have a little video for you to show you how it is that we conduct sampling in the field. Enjoy.
Let's learn the basic water sampling techniques. There are many parameters that can be tested for water quality. Each parameter requires a unique way of collecting samples for testing. Let's take a look at how to collect water samples to be tested for the following parameters: fecal coliform bacteria, biochemical oxygen demand, nitrates, phosphates, suspended and dissolved solids. Water for bacteria testing is always collected first in a clean, sterilized bottle. Be careful not to touch the mouth or inside the cap of the bottle to prevent contamination as you dip once to collect your sample. No rinsing. Fill the bottle only to the shoulder so that the bacteria population can multiply for testing. For biochemical oxygen demand, we ensure that we condition our bottles by rinsing 3 times before collecting the sample. This removes any residue which may have been left behind during the wash process. Carefully and slowly, invert the bottle underwater to reduce bubbles forming during the collection. We want an airtight seal to know exactly how much dissolved oxygen is present in the sample. Try not to collect the sample in turbulent areas, and always remember to fill the bottle all the way to the brim. Our third collection is water for testing nitrates, phosphates, suspended and dissolved solids. Like our biochemical oxygen demand sample, the bottle is rinsed 3 times in the body of water before collection of the final sample. Remember to always discard the rinsed water where it will flow away from the sample point. Here are some additional tips for successful sampling. After collecting your sample for biochemical oxygen demand testing, look to see if there are any bubbles in the bottle. To get rid of these bubbles, gently tap the sides and the mouth of the bottle, and this will cause the bubbles to float out to the top. And for a final tip, always remember, safety first, safety second, and safety third. Let's learn the basic water sampling techniques. There are many parameters that can be tested for water quality. Each parameter requires a unique way of collecting samples for testing. Let's take a look at how to collect water samples to be tested for the following parameters: fecal coliform bacteria, biochemical oxygen demand, nitrates, phosphates, suspended and dissolved solids. Water for bacteria testing is always collected first in a clean, sterilized bottle. Be careful not to touch the mouth or inside the cap of the bottle to prevent contamination as you dip once to collect your sample. No rinsing. Fill the bottle only to the shoulder so that the bacteria population can multiply for testing. For biochemical oxygen demand, we ensure that we condition our bottles by rinsing 3 times before collecting the sample. This removes any residue which may have been left behind during the wash process. Carefully and slowly, invert the bottle underwater to reduce bubbles forming during the collection. We want an airtight seal to know exactly how much dissolved oxygen is present in the sample. Try not to collect the sample in turbulent areas, and always remember to fill the bottle all the way to the brim. Our third collection is water for testing nitrates, phosphates, suspended and dissolved solids. Like our biochemical oxygen demand sample, the bottle is rinsed 3 times in the body of water before collection of the final sample. Remember to always discard the rinsed water where it will flow away from the sample point. Here are some additional tips for successful sampling. After collecting your sample for biochemical oxygen demand testing, look to see if there are any bubbles in the bottle. To get rid of these bubbles, gently tap the sides and the mouth of the bottle, and this will cause the bubbles to float out to the top. And for a final tip, always remember, safety first, safety second, and safety third.
So, okay, I hope you would have gotten some useful information in terms of what you should do if it is that you need to collect samples for your SBAs. Now, before we move on, I have at least one more question based on Mrs. Roden's presentation. And so the first question is, what is the difference between point and non-point sources of pollution? So remember to use the raise your hand button. Jodianne, because Jodianne, you may go ahead, please indicate which school you're from, Jodianne.
I am from St. Catherine High School.
Okay, Jodianne, go ahead.
Okay, so the non-point sources would be sources where, well, unknown sources they can't find, like runoffs after rain. And sources, the point sources would be like drains, gullies, chips, oil spills, and other sources you can find.
Okay, and Safia, she's correct? Okay, wonderful. Congratulations, Jodianne. So you can message the host with your contact information, and we will ensure that you get your prize.
Wait, when you say message, you mean in the chat?
In the chat, message the host in the chat.
Okay.
And we have one more question. The question is, why do we sample? Anyone for that question? Why do we go ahead? Casey? Okay, Casey. Is this the Casey that won before?
Yes, miss. Yes, miss.
You're on a roll.
Miss, we sample because we want to get achievable sample, miss, and reliable, miss.
Okay, Casey, could you again achievable?
What you said, miss?
Could you repeat your answer again, please?
We sample because we want to get achievable and reliable samples.
Yes, I think what we really wanted is reliable samples represent the whole system. But I think you can still get your prize. So just as before, we would have had your contact information, message the post in the chat, and we will make contact with you in terms of your prize. So we will move now to a very short break. We will show you two videos, one on coral reef and one on our environmental club. And so that will give you a chance to take a short break, fresh yourself, and prepare for our next presentation, which would be on coral reefs.
A coral reef. It can be one of the liveliest places on Earth. But what's really surprising is how much more goes on here than meets the eye. Even the corals themselves are alive. They're made up of tiny animals called coral polyps, distant cousins of jellyfish. Hundreds, even thousands of polyps can make up a coral. And this impressive reef, they built it, millimeter by millimeter. How? A polyp is mostly stomach, with a mouth on top. It sits in a hard skeleton that's part of the reef. The polyp takes in dissolved minerals from the water and mixes them with proteins. Then it rises up out of its skeleton, leaving space below. It deposits calcium carbonate, also known as limestone, into this space. Over time, each little polyp not only builds its own skeleton, but adds to the structure of the reef. So what gives the polyp the energy for all this building? A unique partnership. Inside most reef-building polyps are tiny algae called zooxanthellae. They use energy from the sun to photosynthesize, producing up to 95% of the food the corals need. As animals, the corals emit nutrients valuable to the algae too, and also give them a protected place in which to live. This trade-off benefits both the algae and the corals. It's called a symbiotic relationship. The algae are even responsible for the colors we identify with corals.
And I'm the Shadow Vice President of the Jonathan Grant High School's Environmental Club. My thoughts about the program, it is a very good initiative for our schools, the different schools in Region 6, because it will enable us to have better connections with other companies that we wouldn't know how to do per se, but with their help we can. My hope for my club going forward is that we will have a better space for ourselves, because right now we are using a classroom, which on the morning shift, like if we decorate it, they will tear it down or things like that. So my hope for the club in the future is that we will gain more experience, more exposure to things like these, the programs and seminars like these, and also to help other students at my school see that our club is a great club.
My name is Brian Reed from B. Glamere High School. The program will help environmental club by giving them guidance and show them methods and ways and how they can develop the club and help the environment. My hope for my club at my school to go forward is that I would like to see more students in the club, more people participating in the club, etc. Thank you.
Okay. So I hope you would have gotten some information about our environmental club, as well as gotten some preparation for our next presentation. But before we go to our next presentation, we want to hear from you. So a poll should come up on our screen shortly, and we ask as much persons as possible to respond to the poll. We want to hear what your thoughts are in terms of the three presentations that would have just gone before. All right, so while we wait on that to come up, I will move into introducing. So our next presenter is Gabrielle J. Watson, and she has worked at the National Environment and Planning Agency for 5.5 years and spent close to 3 years working with the Jamaica Conservation and Development Trust in managing the Blue and Drunken Mountains National Park and World Heritage Site. She attained her Bachelor's of Science degree in Marine Biology and a Master's of Science degree in Natural Resource Management from the University of the West Indies. She holds a professional certificate in project management from Project Management International, and her presentation will be on the status of Jamaica's coral reefs.
Good morning, everyone. As you heard, I'm Gabrielle J. Watson from the Ecosystems Management Branch at NEPA. And now we're going to get into a little bit of information about the status of Jamaica's reefs. And we're going to start with the general information on coral reefs. We look at the reef monitoring program that NEPA has, as well as the sea surface temperature monitoring program that we have. So some general information on coral reefs, just to give you a brief introduction. Coral reefs value at somewhere around 2.7 trillion US dollars in terms of the protective services, the biodiversity, and the general benefits that are provided to mankind. And that's a nice little infographic that you can use for your information when you're compiling your SBAs. So many of you may not be familiar with what a coral reef looks like. And it may be strange to you that I have there that they're similar to jellyfish. And the reason for this is because they're both in the phylum Cnidaria, so they're like cousins. And looking at it, you wouldn't tell, but they're actually related. Now, coral reefs are actually made up of a calcium carbonate skeleton. And in that calcium carbonate skeleton, there are polyps. And keep with me, go with those polyps now. And on those polyps, there are tentacles, similar to jellyfish that have tentacles. And in those tentacles, you have different cells. So you have one that is a stinging cell, like the jellyfish, and those are called nematocysts. So if you touch it, it might give you a little sting or a little rash. And then there's also the zooxanthellae that you would have heard about in the previous video. The zooxanthellae allow the coral to feed because they photosynthesize, and that means that they are photosynthetic. And these are symbiotic algae that live in the coral polyps that harvest energy from the sun. Now, I mentioned before the different value to coral reefs, and coral reefs, as you can see, are rich in biodiversity. They contribute to economic productivity. They provide a nursery and habitat for marine organisms, similar to the mangroves that you would have heard before, and they act as barriers, protecting the coastline from high storm and wave energy, similar to the mangroves as well. So this is a picture. I mentioned the nematocysts and the zooxanthellae. You can see the calcium carbonate skeleton and the different tentacles. That's just so you have an idea of what a coral reef looks like when you get down to scale. So coral has different growth forms. In looking at the morphology, there are two types of coral. You have your hard coral, also called your stony coral, and these are responsible for building your reefs. They're called reef builders, and they contribute to beaches. They give you sand in those white sandy beaches that you like to go to. Those are part of coral's contribution. And those take different forms. They can be branching, encrusting, have a platy or leafy morphology, or a boulder or a massive morphology. And then there's soft corals, which some of you may be familiar with from seeing them in fish tanks or aquarium. And that's like a sea fan. And if you look at the image in the bottom right corner, and it looks like a fan, almost like a palm, that is a soft coral. Now, coral reefs have different formations. There are four main kinds of formations that a reef structure will have in the sea. The fringing reef is the most common type, and that's what we predominantly have here in Jamaica. And that one is near shore, and it's separated from the landmass by a narrow and shallow lagoon or area of water. Sorry about that. Then there's also the barrier reef, which is similar to the fringing reef, but found further offshore. And that one is separated by a much wider and deeper lagoon or area of water. The atolls are not so common, and you're probably not going to be familiar with seeing that one. They're found often in the middle of the sea. It's essentially a ring, and within that ring, you have a area of water. So it's almost like a pool, like a private pool, sort of. And then patch reefs, we have those here as well, and those are, as it says, patchy. They're isolated, and they tend to be fairly near the shore. So in terms of the geographical distribution of coral reefs, they're generally limited to a particular range because of the requirements that they need to thrive, not just survive, but to thrive. So the geographical distribution is essentially between the Tropic of Cancer and the Tropic of Capricorn. So that's the tropical and semi-tropical regions. And that's around 23 degrees north and south of the equator. And in Jamaica, the coral reef distribution across the island, the coastline is spanned by roughly 1,240 kilometers squared of coral reefs. And as I mentioned before, it's fringing and patch reefs that we have, and those occur mostly along the north and east coast, and to the south on the broader shelf. So I mentioned that there is a specific geographical restriction for coral reefs. Now there's also other conditions necessary for corals to thrive. You would have heard about the mangroves, and you would have heard about water quality. So these things contribute to coral reef development as well. The mangrove presentation spoke about the zonation, and if you recall the presentation that Delroy made, it showed coral reef, seagrass beds, and then you got into the terrestrial area, which was the mangroves. So keep those things in mind. It will be important on this specific slide. So looking at the water temperature, the geographical restriction was between the tropics and semi-tropical areas. And the temperature within that range is roughly between 23 and 29 degrees Celsius. So corals can't survive in cold water. Light. The water quality presentation spoke to light penetration. So light is necessary for corals as well. If the water doesn't have any light coming through, so if it's high nutrient load, there's no water, there's no light penetrating, the coral reef will not be able to photosynthesize. If it can't photosynthesize, it's going to starve. And I'll get into that a little later, but when corals are not able to photosynthesize, they starve, they begin to die, and they lose their zooxanthellae. Clean water. That means it's low in nutrient and low in sedimentation. And sedimentation was also raised in the previous presentation on water quality, and that's essentially silt and excessive sand and small particulate matter that runs off into the water, often from land-based sources or other aspects of point pollution. Flowing water is necessary. It helps to keep things like the sedimentation from covering and stifling the coral that are trying to grow. Salinity. Corals grow in salt water, and typical range of the salinity is from 32 to 42 parts per thousand. Seawater generally around 33 to 35, and it can go higher to more saline areas. And similarly with pH, corals are restricted in their pH tolerance, primarily because of their calcium carbonate skeleton. Calcium carbonate is eroded by acid. So if the pH drops below 8, it's going into a more acidic condition, which will not only erode the calcium carbonate skeleton, but will prevent it from growing. So you'll get very, very thin coral that becomes very brittle, easily damaged, easily broken. depth is restricted to 150 to about, it can go deeper than that, but about 150 feet, which is around 45 meters. And that is similar to accessing light. So the deeper the coral area is, the harder it is for the light to reach to that coral. So coral are generally concentrated within depths within the 150-foot range and above. So you can have coral in very shallow waters, maybe 3 feet of water, to as far as 150. And then of course, a smooth and stable surface for the larvae to attach. If there isn't anywhere for them to attach, then it's not going to grow. If it keeps moving, it's not going to grow. So where you have reef growth and reef development, you can have coral starting to grow on rocks. You can have coral growing on pylons and jetties and other structures that have been built or left in the sea for some extended periods of time. And often those things contribute to artificial reef structures. So I mentioned the conditions necessary for coral reef formation because hard corals are very susceptible to disease and changes in sea temperature and water quality. When they experience any drastic changes in the conditions in the environment in which they're growing, their response is to eject or expel their symbiotic algae. Those symbiotic algae that I mentioned before, zooxanthellae, that are responsible for the photosynthesis, those will leave their body. And once that happens, the coral turns white. And that process is called coral bleaching. So bleaching is when the zooxanthellae or the symbiotic algae is expelled from the coral structure. And if a coral remains in a bleached condition for extended periods of time, it will starve. And if it starves, it will die. So some of the causes of coral reef degradation. So these are threats or challenges to coral reef growth. We mentioned sedimentation. We spoke about temperature, water pollution, and that was extensively discussed prior. So improper waste disposal, whether it be sewage or solid waste and other forms of land-based runoff that could be agricultural waste or stormwater. Physical damage from collisions or boat anchors or even improper snorkeling or diving activities that are from an untrained or inexperienced diver. So the image at the top is showing a damaged reef from a ship collision, and the lower picture is a diseased coral. In terms of overharvesting and unsustainable fishing practices, you often hear that Jamaica is has one of the most overfished waters in the Caribbean, and not just the Caribbean, but in the world, as well as unsustainable fishing practices. And that can range from the method that you're using, whether it be a mesh size that is too small for your fish or your fish pot or your nets, or it could be something like throwing contaminants into the water, something like a poison or a toxin to shock your fish so you can catch them all at one time. Or something a little more, or a little harder to detect is fishing out of season or fishing the wrong size of the animal or the fish that you're looking for. So looking at catching baby fish or fish that are not at the appropriate maturity to be harvested. Disease and, of course, marine and coastal development was mentioned before. And that includes things like dredging for ship channels and port areas. It includes reclamation of wetland areas such as your mangroves for development of hotels, housing developments, creation of beaches and other forms of structures, and things like building out groins and jetties that will have different implications. So in terms of the consequences of these actions, there is a range, and of course these are not an exhaustive list. This is not an exhaustive list, neither was the previous one. So things will include a decrease in biodiversity. You know that bio refers to life and diversity means the variation. So if you have no coral reefs or you have unhealthy coral reefs, you're not going to have much variation. There won't be much life and it essentially will not thrive. You will have a decrease in seafood availability, and anyone who is vegetarian, pescatarian, or just generally likes to go to Hellshire and have some fish, you know that it may not be as easy to get the kind of fish you want, the size of fish you want. And if you look at the picture on the slide, that is a current picture of Hellshire Beach. And you're seeing that the structures are essentially in the water. In terms of beach maintenance, and that's natural beach maintenance and sand replenishment, I mentioned before that corals contribute to the sand that you see on the beach, that makes up the beach. So if you don't have any coral, you lose the sand. The sand, if it's gone offshore, will not come back to the beach. In terms of your coastal vulnerability and storm activity, if you have no protective areas in your coastal environment, then you're exposed, you're vulnerable, and you may not be able to recover readily. And similarly with flooding. When you don't have these coastal protections, there is an increase in the flooding that happens onshore, and it also contributes to saline intrusion. So your groundwater will be contaminated. And then of course, I mentioned white sand beaches. It's aesthetically pleasing. We like to enjoy it, and tourists of course like to enjoy it, and that contributes to our economy. So some of the measures in place to help with coral reef protection and to reduce the impacts. Firstly, we have to look at legislation. Coming from a regulatory standpoint, the National Environment and Planning Agency is a regulatory body. And so we look at different legislation, one of which is the Natural Resources Conservation Authority Act. And under that act, there are various regulations and various orders. For example, you heard about the applications process, and that goes through permits and licenses. So any form of development activity that happens will be subject to the permits and licenses regulations, as well as the Town and Country Planning Act Parish Development Order. You heard about the wastewater and sludge regulations, and these are all plastic ban, which is the plastic packaging materials prohibition order, and these are all things to help to improve the conditions of our environment and reduce the impacts that are occurring. Also, you have the Wildlife Protection Act, the Beach Control Act, the Fisheries Act, and then there are various international treaties, for example, the Convention on Biological Diversity, the Convention on Endangered Trade, on the International Trade of Endangered Species, and marine pollution. Some of the measures to reduce impacts, include zoning of protected areas or designation of protected areas and zoning of areas for different kinds of activity, monitoring enforcement, and different projects to improve the conditions. So on the pictures on the side, you will see a lime coral nursery and invasive species control, that's the lionfish. And of course, public education is very important. So looking at NEPA's Coral Reef Monitoring Program, there are essentially 4 healthy reef indicators that we use, and these are coral cover, macroalgal cover, herbivorous fish like parrotfish, and commercially important fish, which are like your snappers. And this helps us to track the conditions of the reef, to help to gauge the protections that are in place and whether or not they're being considered effective, as well as to improve the measurements and the measures that are in place for adaptive management. So there's a little scale there that shows you what the different ranks are in terms of what we see. So for 2019, our coral reefs were running, and you see anywhere you see orange, it means that it is poor. Anywhere you see red, it means that it is in critical condition. Yellow means it is fair. And if you see green, that means we're doing okay, good to very good. And 66% of our reefs in 2019 of the monitoring sites that we had were ranked as poor. These are the sites that were monitored, and you'll see a lot of red, a lot of orange, and some yellow, but no green. Those are all the 2019 monitoring sites. So based on the monitoring that we have had since roughly 2011, we have continually been ranked in a poor position for our reef condition. So while some parameters like the hard coral and the algae may not be at that stage as yet, it's really the condition of our fish that is contributing to this very low ranking. So you'll see that we are severely overfished, both in herbivorous fish as well as commercial fish. As you can see, a lot of red has been coming up, and it has been worsening. So looking at our sea surface temperature monitoring, and this is the last section, don't worry guys. Sea surface temperature is looking at the correlation, or what we're using it for is look at the correlation with changes in temperature in our waters and the connection to bleaching events. And they started in 2013, we have 27 sites. So essentially any temperatures that are experienced for extended periods that are above 30 degrees Celsius, these can be are more susceptible to bleaching. So we have, these are the 27 sites and the locations that we monitor for sea surface temperature, and it's easier to see here. We have Negril Environment Protection Area, Montego Bay Marine Park, we have the Ocho Rios Marine Park, and that's over by Discovery Bay, you see there. East Portland Fish Sanctuary, Port Royal, which is Palisades Port Royal Protected Area, and Portland Light Protected Area. So for the last monitoring period that we have, this is showing January 2018 to October 2019. And one of our challenges continues to be our inability to retrieve data from the devices that we use. The picture on the first slide for the sea surface temperature showed a small plastic monitor that we attach to reefs, and sometimes we're not able to retrieve the data from those in case of dislodgement or flooding of the device. However, you will see from this data that there are some cells showing here that are highlighted in red, and those are exceeding the threshold. So that's in Montego Bay Marine Park, which had a maximum temperature over 30. Similarly, Pear Tree Bottom in Discovery Bay had a maximum temperature of over 30, and both of those averaged temperatures exceeding 30 for that period. So when we retrieve our next set of data, we can compare to see if it is an extended period of time that those temperatures are being observed and whether or not it is having an impact on an increase in bleaching and susceptibility to the corals. So this is the last slide, and you will see that it's showing the warmest temperatures, or the months having the warmest temperatures, and this is concentrated between June and November. With the anomaly showing in 2019 for February, and that was at a site in Discovery Bay, the concentration of high temperatures is really between June and November, and that aligns with our Atlantic hurricane season. So you know that warm seas give you hurricanes. So between June and November is where we have seen the highest temperatures recorded. And that is the end of this presentation. Some references if you need them. And I have 2 questions for you. So the first question I am going to ask is, what is coral bleaching? And you would have heard it in the video as well as in my presentation. Giovanni Clark, can I hear your response?
Coral bleaching is when the symbiotic algae leaves the coral structure, causing it to have a white color, and resulting in, if this continues for a long period, it will result in the coral to die.
Very good response. Thank you. It's Giovanni Clark. Thank you, and reach out to the host in the chat so that you can get your prize. And a second question, can you name 2 conditions necessary for coral reef growth and development? Any 2? Jada Lynn Henry, can I hear from you?
Water temperature, and it must be 23 to 29 degrees, and clean water, which.
And what does clean water mean?
It should be low. It's not the water should not be polluted, and it should be low in
Okay, we'll take it, Jada. All right, and sorry, Jada. Yes, it's Jada Lynn Henry. Thank you, Jada, for your participation, and thank you for being so attentive.
Thank you so much, Gabriel J. Do we have any questions for Ms. Watson? No question. Rachel Blake. Okay, Rachel, you have a question for Ms. Watson?
Hi,
Rachel?
yes, I do. Sorry, I don't know why I wasn't getting to unmute. Um, I have a question for pertaining to coral reefs. Can they be like after they're bleached? Can they come back to life?
Hi, Rachel. That's an excellent question. Yes, bleached corals can recover if they are not bleached for extended periods of time or if they're not extensively bleached. So if you look at bleaching, it tends to start in one section of the coral and then it will expand depending on how long and how exposed they are to those conditions. So they can recover if it's not too long of a period or too extensive. They don't have to die once they're bleached.
Okay, one more question. You said that they need light for photosynthesis. Were you like saying that as if you had some corals that weren't reaching the light, like wasn't getting to penetrate on them? And what can cover them in order, like, is there a ship, would there, if, just saying like a ship would be there and light would be able to get in? Like what can be on the surface to not allow light to reach a coral?
So in terms of light penetration, it's two things. One, I mentioned the clarity of the water. So if the water is not clear, if it's not clean water, it means that it's being blocked by, it could be extensive pollution on the top. The water could be dark in color, so the light is being blocked and being absorbed by the different elements on the surface of the water. It could be garbage, it could be other kinds of runoff, it could be oil, it could be anything like that that would block the light from reaching, as well as construction that's done over the sea. So coral can grow in very shallow waters, and sometimes you will see structures that are built over that area. If coral are there and they're not able to get light from spaces between those structures, the coral may not be able to survive because of the absence of light, as well as the actual depth. I mentioned that there is a depth restriction to coral growth, and if it's too far, particularly the deeper you go, the harder it is for light to reach to those areas without having anything being on the surface. Just the fact that it is so much further down deeper in depth, the light would not be able to penetrate. So construction over the sea, water clarity itself, as well as the depth. Is that clear? Rachel?
Yes, Miss. Thank you so much. I enjoyed your presentation. Thank you for answering my questions.
Okay, thank you, Rachel. Before we move into our next presentation, we have one more question from Jodianne. Jodianne, go ahead.
Yes, my question is, what can be done to restore or protect Jamaica's coral reefs?
All right. When you get the presentations later on, you'll be able to see that some of the measures were outlined, and they range from legislation to different projects. And I showed on one of the slides a picture of a coral nursery. So you can have rehabilitation for coral areas, reef restoration. You can have restoration of your terrestrial areas, such as wetland restoration. A lot of mangrove planting activities do happen. There are I mentioned the different legislation. So ensuring that all developments are done within the conditions of permits and approvals that are granted. And of course, general community stewardship and participating and paying attention to the different sensitization sessions and making opportunity to participate in different training initiatives, as well as sharing the information that you learn with those around you. So there are various things that can be done, from a small scale to a large scale.
All right, thank you, Jodianne. So we will also reserve some time at the end of the presentation for additional questions. So now we're going to move into our final presentation for this morning. And our final presenter is Mrs. Kenisha Brown Russell. She is the head of Department of Geography and Dean of Academic Studies at the Camperdown High School, where she has been teaching for 17 years. She is also president of the Geography Teachers Association and supervisor of the Cape SBA for CXC. Over the years, Mrs. Russell has worked assiduously to improve the achievements of students in geography at both the CSEC and the Cape level. So Mrs. Russell will go into giving you some general SBA tips and guidelines as you seek to make your preparations. Mrs. Russell.
Thank you so much, Ms. Robinson, and good morning, students who are at school. Good morning to the students who may be at home, and good morning to the teachers. First and foremost, I want to say a big thank you to the NEPA team for forging ahead with their annual webinar series, because indeed the school-based assessment is such a very critical component of the CSEC examinations. And in light of what is happening now, I know quite a number of students are sitting behind their computers or with their phones, and I know there's some amount of anxiety as it relates to completing those SBAs. And why? We know that geography and environmental science rest heavily on going out into the field to collect data. And given the pandemic situation, I know that a whole lot of persons are thinking that, oh, I can't go out, and the SBA is going to be compromised in some sort of way. But I'm hoping that at the end of the presentation, I'll be able to share some tips with you in terms of how can we go about executing the SBA such that we are able to get a very good grade at the end, because that grade is very important to the final exams. All right, so just before I start, I really want to get an idea of my audience. So I'm going to go ahead and ask the technical team to assist me with this poll. And I want to find out, have you started the SBA? Is it yes or is it no? Have we started? All right, I am thinking that there should be a poll coming up and you click either yes or no. So if you have started SBA, you click yes. If not, you click no. All right, so we give them a little bit more time. All right, we're good. All right, so we're trying to find out if the geography SBA has begun. All right, any results from the technical team? All right, so 78% of the persons who are with us have started. Good, and that's a good place to start. All right, now, if you have started, and for the persons who haven't started as well, what percentage of the SBA have you completed? All right, so is that 0%, and that would be for the persons who haven't started? Is it somewhere at 20% or less than that? Or are we more than 20% complete? Or are we at least 50% complete? It can be more than 50, but our baseline there is 50% at least. Where are we as it relates to the completion of that SBA? All right, so the majority is saying 20% or less. And I am not too daunted by that percent, because the most important thing at this point in time is for us to start. And once we have started, it means that we are going to get to completion long before the due date. And we're supposed to have that due date in our minds. So I'm not getting any response there. Sure, clicking. Oh, okay. All right, so we should be finished before the due date as long as we have started. Now, generally, we would have known that the SBA constitutes 20% of your overall grade. And for me, I call this the cheat sheet for CSEC. And why is it a cheat sheet? You get the opportunity to go into the exam room with 20% before you even start writing that paper 1 or 2. No, we don't want to throw away not even 1% out of that 20. So with the tips that I'm going to give to you, I'm hoping that we will get as close as possible to that 40 marks, as much as possible. All right. Now the geography SBA should be somewhere at 1,000 words, and that excludes the bibliography, that excludes the illustrations, that excludes the appendices. So I know oftentimes we'll be typing the SBA and we're looking on the word count and we're seeing somewhere way over 1,000. Sometimes we have to go back and we have to select the text that actually contributes to that final count. And I mean, if you're seeing 2,000, chances are you may 1,000 maybe geared towards your name, the page number, the titling of the illustrations. All right, so it's very important for us to go and select and make sure that we have a proper count of the SBA. I always say that the SBA makes a difference between a good grade and failing. If you go into the exam with at least with 20% maximum, then chances are you will be walking out with a good grade. It also makes a difference between failing, because we should have known by now, if we don't submit our SBA, then we get an automatic ungraded. So the SBA is very important. Now, as I said, this year may present some amount of challenges, but nonetheless, there are several methods that we can really employ in order to get the work done. Now, if we look at the screen right now, I have extracted just a tiny section from the SBA treatment handbook that was produced by CSEC. And what we're seeing right now is that no major changes were made to the SBA, in that you are not getting the opportunity to just get a grade based on what the teacher may recommend. You have to execute a SBA, you have to submit one. So there are no changes where that is concerned. What has changed is how we go about actually implementing the SBA, and that speaks most to the methodology that is being applied. And I'm going to talk a little bit more as it relates to that as we go on. Now, still from that publication that was done by CSEC, virtual field trips are allowed. And if you browse on the internet, there are so many different types of virtual field trips that will pop up. But for our purposes, we want virtual field trips that allow you to capture the data that you would have gotten out in the field from the traditional field trips. And there are a lot of different modalities that can be used for that. For my own school, we actually employed this method whereby the teachers were the ones who went out in the field. So there were no students on the trip, sad to say. And we went out and we made an attempt to connect with the students via Zoom. So we are at the location and we are actually collecting data while they're watching. And there was quite a bit of participation because they were able to talk about the methodology, they were able to talk about what they were seeing. So it is indeed a very good method that can be employed as it relates to especially the natural sciences component of the SBA. All right, now CXC is also proposing that the field trips are conducted in small groups. The guiding can be done remotely. So whereas the teacher would have been the one out in the field and they're the one saying, doing that. If it is that you have decided to undertake a field study in your own community, then it means that your teacher could be guiding you remotely. So they're on a Zoom call and they're, guiding you along in terms of collecting the data along the way. Another thing too is that your own communities are at your disposal. And there are a whole range of topics that you can explore as it relates to the very community that you live in. All right, still on some COVID considerations, again too, as it relates to the methods that can be employed. I know quite a number of studies, the human studies, rest on the use of questionnaires. And in so doing, it means that we have to collect data from persons. Now, it may be that we can't go out in order to collect that data. So it means that we may have to be utilizing the virtual platforms such as Zoom in order to collect that information. We could be using SurveyMonkey. All right, let me go back to that. Right, so here we are. So we can utilize survey tools such as Google Forms. We can use SurveyMonkey in order to collect that data. We can use social media. So we can actually distribute those questionnaires via WhatsApp or email. So all is not lost at all. And definitely we can make use of Google Earth and Google Maps in order to make some assessment as it relates to the study area, as well as drawing or sketch maps, which is very important, and I'm going to come to that in a bit. All right, now our very own EO, Miss Jasmine Waite, she has gone ahead and she has actually browsed through the syllabus, and she has outlined various different topics that can be used to conduct the field study. All right, those that minimize contact out in the field. And let me just quickly go to that. All right, so we're skipping over here for now. All right, so we are looking at, wow. All right, this is not where I want to be at all. All right, but you have several topics that can be looked at from the syllabus, and that includes population. We can look at drainage basins, and I'm going to look at that in a little bit. You have the flexibility to do some amount of hazard in terms of response, in terms of the impact in a particular community. You also have the flexibility to look at industries. So we're talking about primary industries, we're talking about secondary industries, we're talking about tertiary industries. You have the flexibility to craft a SBA around that topic, and you are actually able to gather data without actually going out into the field. All right, we are at the components of the SBA. All right, scroll up some more. All right, so we're right there. All right, so wow. All right, so let's look at the different components of the SBA. And this is where we get out our books now and our pens and we start writing. For those persons who haven't started, we wanted to have a look at the screen right now. Now, the main components of the SBA, we have the aim, we have the introduction, we have the location of the study area, we have the methodology, we have the presentation of data, we have the analysis of data, and we have the conclusion, and last but not least, we have the bibliography. And if there's an appendix, you can also include that in your study. All right, now I am going to go ahead and we are going to look at each component of the SBA. And that starts with the selection of the topic. Now, what kind of consideration should we be making as it relates to the topic? Now, before I said, the syllabus is our main guide in terms of selecting our topics. So first and foremost, you want to take, if you haven't started the SBA, you want to take out that syllabus and you want to ensure that whatever idea pops in your head, it is outlined in the syllabus as a part of the objective. Very important. The next thing that you want to ask yourself is, am I able to adequately collect data in a particular location? Very important, because if you are not able to do so in a very defined area, then what happens is that the SBA gets too big and bulky and you would not be able to adequately achieve your aim. The next thing too, am I able to formulate a research question based on the topic that I have selected? So very, very important considerations that ought to be made. All right. Now, when we get to selecting that topic, one of the first things that we really want to do is do some background research on that topic. Very important. And oftentimes we tend to skip over that step because we think we know the information, but it's very important to do some background research. So you're researching on the methodology that you want to employ for the SBA. You are researching on general information as it relates to the topic so that you can have some assistance as it relates to your analysis. Now, after doing that research, you want to start formulating your research question, and after which it will be very easy for you to actually formulate your aim. All right, so let's look at those steps. How do we go about selecting that topic? And I'm speaking primarily to those persons who are yet to start the SBA. Right. Sometimes I click, but there's no response. All right. Now I followed the syllabus. I looked through the syllabus and I decided that I am going to look at some topics that are very useful at this time. Some topics that we can definitely collect information, whether it is we're out there in the field or we're at home using the different online mediums to collect information. Now, what are those topics? And I started looking at this earlier. Drainage patterns. You have the option to select a topic based on that. Hazards. So we are looking at impact. You're looking at the response of the hazards in a particular community. So those relate to the natural component of the SBA. All right, now, as it relates to the human systems, we're looking at population dynamics. So here we can look at changes in population. You can analyze your dot maps. You can look at your population pyramids in order to assess changes in population growth over a particular period in a particular location. You also have the option to look at industrial location. So here we're looking at factors influencing industries. So for example, if we're looking at a food processing industry, such as St. Mary Banana Estate, that we know produce banana chips, what are those factors that influence where they are located? Now, based on the syllabus, you have the option to look at any industry that relates to fishing, forestry, bauxite, food processing. So your duty is to ensure that whatever it is that you select, it's in alignment with what is in the syllabus. You can look at the benefits and the problems relating to those industries as well. All right, so Mrs. Russell went ahead and we want to look at the steps now in terms of selecting that topic. So as an example, I selected drainage patterns. All right, so we're looking at drainage patterns. Now, is that topic a part of the syllabus? Definitely. So, because when we look in the syllabus, we realize that yes, drainage pattern is there. Now, what we want to do is we are describing drainage pattern and looking at the relationship to rock type and geology. Now, are we able to conduct such a study in a defined geographical area? Yes, we can, because we know that we have drainage patterns in different drainage basins all over Jamaica, right, NEPA team? And we are able to conduct drainage basin analysis in just about any watershed that we have in Jamaica. Now, to narrow down this research, because this is also a very critical component of the SBA, we are looking at St. Thomas and we're looking at Clarendon. All right, so we want to select one watershed from St. Thomas and we want to select another from Clarendon. All right, so here we have our topic again. We have our location. And in order to narrow down that location, we have decided to choose Yalas Drainage Basin in St. Thomas, and we have selected the Rio Bueno Drainage Basin in Clarendon. So what is it that we want to do next? We want to formulate our research question. So we have our topic, we have our location. But what is our research question? What are we going to be doing for the SBA? So based on our location, one of the research questions that is very plausible is, how does the drainage pattern of the Rio Bueno Drainage Basin differ from that of the Yalas Drainage Basin? Very plausible. Or what are the factors influencing the development of the drainage patterns in the Yalas and the Rio Bueno Drainage Basins? So we have satisfied the criteria of formulating a research question. Now from that, we're able to deduce our aim. And what is our aim? This is very specific and it basically answers the question that we are going to be doing in our research. In order to formulate our aim, we are using descriptive terms such as to identify, to describe, to discuss, to observe, to explain, to compare, to contrast. Very important because the aim is you doing something, so you have to execute. So from that very research question, how can we formulate our aim? All right, so one, we need to use those descriptive terms, and two, we must include the study area in that aim. All right, so let's look at the aims that we came up with. All right, so aim number one, to explain how geology affects the drainage patterns of two drainage basins. Now, if we assess that aim, we would not be getting full marks for it at all because one, the area is not defined enough, and we need a little bit more specificity as it relates to what exactly we are going to be doing. All right, so let's look at aim number two, to explain how geology affects the drainage patterns of the Rio Bueno and the Yalas drainage basins. So definitely we're getting there in terms of the aim because we have to ensure that we have all the components there. All right, so aim number two, much better than aim number one because we have defined our study area. Or an alternative aim is to compare the drainage patterns of the Rio Bueno and the Yalas drainage basins and to explain how geology influenced the development of the patterns. All right, so I can see the NEPA team perking up now because they know that they can assist in this area, right? So we definitely want to formulate our aim. We want it to be narrow enough so that we can collect information and we can actually go about answering that question in 1,000 words. We don't have a lot of words to play with. All right, now on screen, you are seeing some alternative aims, and I took this from the human systems component of the syllabus. So one, you have to identify and describe the main factors influencing the location of the St. Mary banana food processing industry in Annotto Bay, St. Mary. So where did that come from in the syllabus? We're talking about industries and we're looking at locational factors. Another one, to analyze the effectiveness of community members' response to flooding in Taylor's Land in St. Thomas. So here we're looking at hazard. And this could be your own community. And what you want to do is to analyze how effective the community members are in preparing for a flooding event or a hurricane. All right, now, after we have formulated our aim, what is next? What do we need to do? We now need to formulate our methodology. And this part of the component of the SBA is so very important because if we choose the wrong methods, then we will end up with the wrong information. Or we don't have adequate information in order to answer our aim. So a whole lot of time must be spent on trying to develop our methodology. Now, in writing up the methodology in the SBA, there are three little questions that you answer. Where will data be collected? When will data be collected? And how are you going to go about collecting that data? And that third question is very important, and that's the part you now need to research. How am I going to get information for the topic that I have chosen for the SBA? So again, we're going back to research. Now, the topic that you select or the research that you decide to do, chances are somebody on the internet have done it already. It's in a journal, it's in a book, it's in a publication somewhere. So nothing is wrong in actually modifying what you have actually researched and using it for your own purposes. So that research step is very important. Then you want to focus your research methods, and then afterwards it's time to execute your methodology. All right, now what in writing up that methodology, and this is 4 marks for the SBA, so we have to ensure that we have everything. You have to state where you got your primary and secondary data. You have to give a precise location of the study area. You need to state when primary and secondary data was collected. And most importantly, you must describe all the methods that you actually use to collect data. All right? And we want to ensure that that's in a logical sequence. So you start out by saying, okay, first this was done, then this was done, then that was done. All right? So you're writing all of that in order to maximize that 4 marks. Now, let's look at the methodology as it relates to the topic that we selected. So we're back to our drainage basin topic now. Now, how are we going to get information so that we can compare the Yalas drainage basin with the Rio Bueno drainage basin? Now, central to every topic that we select for geography or topographic maps are very essential. We need it for locational analysis, and we definitely also need it in order to draw our sketch maps. So the first step is you sourcing those topo maps. So those big sheet maps that you get in class to find the grid reference or to find the bearing, those are topographic maps, and you need those for your study. Now, in this case, you want to delineate the drainage basin. So if you look at the slide right there, you see a boundary surrounding the drainage basin. So that would be a very important step for you, because you now need to look at a particular area. So you are going to be drawing a boundary around that watershed so that you're able to only look in that defined area. Now, as I said previously, there are several agencies that have the information, have maps that can help you in this regard. So NEPA is one such agency, the Mines and Geology, Water Resource Authority. You can always lean on these agencies to supply you with maps. I mean, you have to give them time to source the information, but nonetheless, I'm sure they will be more than happy to assist. All right? The next thing you need to do is, after you have collected your maps, you now need to determine what your drainage patterns are like in both basins. And after you're done with that, you just need to compare. Compare what is the drainage pattern like in Rio Bueno, what is it like in Yalas, how do they differ, and how does the geology contribute to that difference? So easy cheesy. And I'm going to emphasize again, once you have that methodology outlined, you are able to get the information that you need. But this step cannot be overlooked, all right? I put that there as an example of the topography map, just in case somebody forget what the topo maps look like. I know COVID take a little toll on some persons, but that's our topo map, and you are using it for several different things in the SBA. All right, I'm going to turn a little bit of attention to the human component of the SBA, because oftentimes persons do topics relating to hazards, to industries, and these are topics that rest heavily on a questionnaire design. And As someone who supervises the SBA, oftentimes I find that the questionnaire is not done properly. The questions are not worded properly. The questions that are in the questionnaire, it does not achieve, it doesn't meet the aim, all sorts of things. So what are the considerations that we want for questionnaire decided? One, you first need to decide on the information that you need in order to achieve your aim, because those are the questions that you're going to develop. It must be linked to your aim. You're going to keep it short. You don't want to be asking persons too many questions. So I say a maximum of 10 questions. If you have to go over that, don't go beyond, say, 2 or 3 more questions. Keep the wording simple, because remember, you're going to be interviewing a wide cross-section of persons from society, and you don't want to be spending too much time explaining to them what exactly the question means. All right. Next, you want to ensure that the questions are relevant to the study. And this is very important, because a lot of time we see questions that have nothing to do with the study at all. Next, you want to make sure that the questions are close-ended, because remember, it's the questionnaire that you're going to use to actually produce your illustrations, and the close-ended questions give you that opportunity to do your pie charts, to do your graphs, and to construct your tables. Last but not least, I would love if we could do a test run. Now, after crafting that questionnaire, you probably now need to find somebody in your household or one of your friends and do a little test run. So you ask each other a question just to ensure that the questions are relevant and the wording is good. Another key area of the questionnaire, and that's administering. Who are we going to give this questionnaire to? Who is going to answer the questionnaire? All right, now that relates to our sample size, and this is so important, because oftentimes we select a sample that is way too small for the population that we're researching. So you have a whole community of, say, 500 persons, and you decide to only ask 10 persons from the community questions. Not sufficient at all. Now, as a rule of thumb, I mean, in the research world, you are really supposed to be sampling 10% of the population, but I know that might be a little, maybe too much depending on where you're conducting your research. So I say 5%. So if the population has 500 persons, then we're looking at a sample size of 25 persons, and that's achievable. All right, as long as you make sure that you have enough time to give the questionnaire and get back the responses. All right. The sampling technique is also very important, and I wish I had a little bit more time to spend on choosing the sampling technique, but the technique that you actually choose can skew the information that you get. So for example, you go out in the field and you are actually conducting a study on street vending in a particular town. You want to ensure that you have a rough estimate of the number of persons who are engaged in that particular activity, and from that you are able to actually draw your sample size. So and this is why your background research is so important. So you would now need, for a topic of that nature, you would now need to link with the municipality, statin, any other agency that can actually give you some idea of that population, because you want to ensure that that sample size is as representative as possible. So it's very important. At the bottom of the slide there, I just dropped in 2 different types of sampling technique, 2 that we tend to confuse, and that is convenient sampling and random sampling. Now, convenient sampling is you going on the road, say you're conducting a study on street vending, and you scout out the area and you see a nice old lady that look like, she will actually answer your question. That is you conveniently selecting that person to answer. And that includes a whole lot of bias. And what may happen to is that some of the information that you really need for the study, you don't get an opportunity to get it because your sample is very biased. All right? That differs from a random sample whereby you randomly select persons from that population. And again, in order to effectively do that random sampling, you have to know the number of persons that forms a part of your population. All right, so very, very important. So for the random sample, you can do a random table in order to select. You can tear up bits of paper and say, for example, you have 10 persons, or let me increase that, 50 persons within a population, and you want to sample. You write 1 to 50 on bits of paper, you tear it up, you fold it, you put it in, you drop it in a hat, or you drop it in a box, and you randomly select numbers from that box. So if you select 5, you're going to select, and that person is number 5, you're going to sample that person, the 10th person, and so on. All right, so random sampling really doesn't speak to just going out and looking at the first person and say, okay, I'm going to interview that person. It's actually more than that. All right, so we are at the location of the study, and everybody who is listening should know that the maps are central to the SBA. And for the study, you are going to be including two maps in that research. So one, you have the map of your territory, and in this case, it's Jamaica. And secondly, you have your site map. Now, oftentimes we tend to, we really tend to ignore map number 2. And that is really not good because you will be actually losing marks. So the site map, and let me quickly run to those. Site map is very important, where you are actually looking at your study area relative to any surrounding features such as the roads, the rivers. Yikes. Okay. All right. So these are examples of your locational maps. I don't know why this is turning like this, but that would be your map number 1. This here would be your site map. All right, I'm not so sure why things turning upside down, but all right, but this would be your site map. Now, sometimes we don't maximize the full marks for that SBA, and why? We have central things that are missing. The key is missing, the title is missing, the colors that are used on the map is different from the colors in the key. So things like that you really don't want to be ignoring, so that and you end up losing marks for the location on maps. All right. All right. So this map here is possibly a good illustration of what we really want when we say map number 2. So we're looking at the study area, and we're ensuring that all the essential features of a map are included on the page. All right, I'm going to quickly run to the presentation of data. Now, this is the bulk of your SBA, the presentation of data and the analysis. We are talking about roughly 50% of the total marks for the SBA. This is where you just get into writing up the findings that you got from your questionnaire, the findings that you got from your map, the findings that you got from your interview. All of those, you're actually putting that together with the use of illustrations in the study. Now, there are 2 methods that you can employ as it relates to the presentation of data and the analysis. One, you can either write up the presentation of data separate from the analysis, or you can seek to integrate the 2. The method that you choose is entirely up to you. All right. Now, as I said, the presentation of data includes various types of illustrations. All right. So we're talking about our pie charts or bar graphs, or, all right. Let's wait for it to respond. All right, so we're talking about our pie charts, or graphs, or tables, or photographs, or sketches. And a lot of times we include these in our SBA, but several things are missing. And because of that, we end up we don't get full marks. So things like titling the SBA, titling the illustration is very important. Ensuring that there's a key for the sketch map, ensuring that there's a caption for the different axes. All right, and we're going to spend a little time, just a minute, to look at some of these. Now, if we look at this pie chart, a lot of time we'll get pie charts looking like this. All right, sometimes there's no title, and there are no percentages on the pie chart. So you end up losing marks. So please ensure that every time you construct a pie chart, the percentages are included, the key is there, the title is there. Let's look on another one. All right, I put this pie chart here, and why? When we look at that pie chart, we see a big circle, and all of that circle relates to yes. Now, a lot of times we get pie charts looking like this. And why is it incorrect? Remember, pie charts actually show parts of a whole. So you're looking at proportion. So if you actually do your questionnaires and you find that everybody said yes from the sample, then there's absolutely no reason for you to illustrate that. If it's a yes, everybody, then it's a yes. But using a pie chart to show it would not be very relevant at all. Let's look at the bar graph. A lot of times we get bar graphs looking like this. And you think it's fine, but it's not at all. For bar graphs, we definitely need a title. You need to ensure that the axes are labeled. So you must label the y-axis, you must label the x-axis in order to get full marks. And that caption at the bottom is very important. So all the illustrations that you have in the SBA must be labeled, figure 1, figure 2, figure 3, and a short caption to say exactly what it is. Very important. Last but not least, our photographs. These are very important too. Sometimes we get photographs looking like this. You produce photographs. We don't know what you're pointing to. There are no labels. So very importantly, you have to ensure that all the annotations are placed on the photograph. And all the illustrations are actually within a border as well. All right? And that caption at the bottom, very, very important. All right, now here is a diagram, and I have a big X on the side there. Why is this not appropriate? This is a diagram that was taken from the internet. And as it relates to the SBA, you are not allowed to copy diagrams from the net or take it from a textbook. Any diagram that you put in the SBA must be yours. You drew it yourself. So even if you see something that you possibly want from the internet, it is your duty to modify it so that it best suits your research. All right, now I'm going to just browse over this a little bit because time is going now. So the presentation of data, as we said, we want to ensure that there's integration of both the text and illustrations. Far too often, you end up producing an SBA where you have all the illustrations on a few pages, and then you have all the text or the description on another set of pages. What you want to do is to marry both of them, put both together. So you must have a continuous description with illustrations interwoven between. All right, so we really don't want to forget that at all. All right. All right, so those are the pitfalls. A lot of times we don't use the illustrations, we leave out those labels. Very important. We have to ensure that we have everything in order to capture that full marks. All right. Now, our conclusion. So we are at the end of the SBA. This is 4 marks, and a lot of times we don't pick up the 4 marks. And why? we have a conclusion that has nothing to do with the aim that we started out with, and or the conclusion doesn't adequately summarize the research. So when you're writing your conclusion, you're going to ensure that one, it's a summary of all the presentation of data and the analysis that you did in the study. You're going to ensure that those points that you put in the summary relates to your aim. And also, especially for the human studies, you're allowed to write some sort of solution or any limitation that you may have in conducting the study. So your conclusions should look, you know, something probably like 3/4 page, half page to 3/4 page, if it is that you are supposed to summarize adequately. All right. Now, definitely put this here intentionally, because a lot of times we write conclusions like this. We say that the aim was achieved or the SBA was a success, and we feel good because you're concluding to say that yes, everything was okay and everything was achieved. But please remember that the conclusion is actually a summary of what you actually did in the SBA. All right. Now, last, we're winding down our bibliography. All right. Now, our bib is a compilation of texts, websites, and other sources used in the study. Now, you have two main formats that you can use to actually cite these books. You have the MLA style, the APA style, whichever one you prefer, that's okay. But most importantly, you are supposed to cite the books first, and then the websites, the maps, the newspaper clippings after those books. All right, so here is an example. So in alphabetical order, by author's last name, you start with the books, and forgive me, that should have been aligned a little bit more, the name of the text that is. So you have the author's last name, followed by a comma, the initial of the first name. Then you have the title of the book, the publisher, place where it's published, and the year. So all those books are listed first. Thereafter, you put all the other sources that you use throughout the SBA. So here we have the map. We used a sheet map, and we used a website in order to execute our study. Now, what is important too, all those books that you used in the study, you must make reference to them somewhere in the SBA. So I don't want at the end of the SBA say, oh geez, I need to find 2 books. You would have been relying on the use of it for your analysis. So please ensure that somewhere in that presentation of data and analysis, you would have made some sort of reference to those books. All right. Other components of the SBA, the introduction, and why do I have it last? Sometimes it makes sense to write the intro last, because after completing the SBA, we may have gone in a different direction, and we want to ensure that the introduction adequately captures what we did in the study. So there are 2 things that you need in that introduction. You must justify why you're doing the study, and you must give a reason for selecting that particular location. Very, very, very important. So that 2 marks is very important to the SBA. And last but not least, you have your appendix. And what do we include in the appendix? If you use that questionnaire to conduct your study, then you include a copy of it, a clean copy that is, not one that you actually wrote on. Any photographs that were central to the data collection methods, you can include that. It's not very necessary, but you can, as well as any statistical information that you got from secondary sources. So if it is that you got info that was very important, say, for example, data from Statistical Institute of Jamaica relating to population, and you use that as your analysis in the study, then you can put that raw data in the appendix. All right, now, did I go over time? Well, not really. Students, I know that was a whole lot, but based on what was presented, I'm hoping that you would have used everything to either improve what you have had so far, or to get started, and that's very important. Even though you may have up until March to complete the SBA, time is running quickly, and it's very, very important for you to start right now. All right, so I am going to take 2 questions. Is that all right? All right, so 2 questions before I ask a question of my own. All right. Okay. All right, so while we wait on the questions to come in, I want anybody to tell me what are 2 important things that you must do as it relates to the illustrations that you put in the SBA. 2 very important things that you must do. All right, so Jodianne, you can go ahead. Let me hear you.
Good afternoon. When you say illustrations, you mean pictures and stuff, right?
We're talking about the graphs, we're talking about the photographs. Yes, so that's what we're referring to.
Well, what I've learned from school, when you're placing the photographs, they should be known as plates, correct? And for illustration, you must always put in key details like keys and legends. It must be bordered, it must be done neatly, the information should be there. So yeah, that's what I learned.
Very good. And you actually touched on an area that I didn't include in the presentation, more so because it's really nuances as it relates to the labeling of photographs versus other illustrations. I know some teachers would have said to you that you can label your photographs as plates, and the other illustrations you label it as figures. I am quite certain that you're not penalized for labeling your photograph as figures as well, and or if it's split. So it's just a matter of choice. But thanks, Jodianne, you're very correct. All right, and there are no questions? All right, so it means that everything is crystal, and we are going to be producing SBAs that get 40 out of 40. So we have 1 question. Brian? All right, so I didn't get the first name. Brianna. All right. All right, so Brianna Lawrence. All right, so you can.
Good afternoon. Would this presentation be sent to us individually?
Yes, I am quite sure that's what the NEPA organizer said. So you will get the presentation for your own purposes.
Okay, thank you.
All right, so thank you very much for staying with me. And I do hope that come March, we will be submitting some very lovely SBAs. All right, thank you much.
Okay, thank you so much, Mrs. Russell. And I'm hoping that having gotten the technical information that you needed in the sessions that gone before and the guidelines from Mrs. Russell, that we will see much improved grades in geography, and that you can actually go into your examination with as close to 20 marks as is required so that we can get good grades. Now, if I want to throw out, because we do have a little bit more time, if there are any general questions, we have with us Mrs. Roden, and she did water quality, Ms. Watson that did coral reef, and we had Mrs. Russell as well. So if there's those persons are on site, so if you have specific questions for any one of those presenters, now is the time for you to ask that question. So are there any questions at this point? Okay, so there's no questions, so that means that one person. Rachel Blake. Okay, Rachel, do you have a question? Rachel, could you indicate who the question is for?
Well, the question is for the NEPA department. So Miss, I don't remember her name. Someone made reference to the fact that we can get resources from NEPA, which can aid in our research. How do we get them? Do we have to visit the office? Is there an online platform for us to visit and speak to someone?
So I believe Ms. Miller had touched on this in her presentation. So usually, normal circumstances, persons can call and make an appointment and come in physically to use our library facilities. As it stands now, we have COVID restrictions, so we are limited to the number of persons that we can access in-house at any one time. I think now we can only accommodate 2 persons at a time in office, and we need to make an appointment. Outside of that, though, you can contact us via email. The emails that we had mentioned before is pubed@nepa.gov.jm, so that's pubed@nepa.gov.jm, or you can also reach out to the library specifically via Doc Center, DOCENTRE, on screen now, .jm, and you can indicate specifically what it is that you want. If the information is in soft copy and we are able to, we will send that via email. If you're doing an interview, the same process applies. We ask that you send your questions beforehand, and then we try to facilitate an interview with a technical officer. So there are opportunities for you to access the resources, and we try to make accommodations as best as possible in terms of COVID-19.
Okay, you think? Sorry, go ahead.
No, I was saying, does that answer your question?
Oh, yes. Thank you so much for answering my question, by the way. All right, thank you.
Is there any? Okay, Ms. Manderson, you can go ahead.
Good evening. This question is aimed at the lady that presented on the coral reef. So she in one of her, when she was answering one of the questions, she mentioned the coral reef nursery. So my question is, what exactly is the objectives and aim of the coral reef nursery?
Okay, so Ms. Watson will be able to respond to that question.
Good afternoon, Ms. Anderson. Thank you for your question. Okay, a coral reef nursery is essentially, it's like a gardening method. You know, if you've been to a greenhouse or you have been to a plant nursery, you see that there are young plants that you grow in a specific environment. You're controlling their growth, you're controlling the conditions around them, and you're monitoring them constantly to ensure that they reach to a stage of some independence before you're able to plant them out into a wider space. So there are coral nurseries that can be in situ. That means they're in the environment that they would naturally occur in. So you have a coral nursery. The one on my slide was showing a line nursery within the marine environment. And that nursery is, you have young corals, and that was asexual reproduction that is using fragmentation. So coral pieces are hung and grown, cleaned and monitored daily until they reach, for about a year, until they're at a size that they can be outplanted onto a larger reef and environment or onto an artificial reef. Coral nurseries that are done ex situ, those are usually done in a tank environment, and those are more streamlined conditions because you can control the water quality and so on. However, when you do nurseries in an in situ environment, the survival is usually a little better because it is mimicking or it's actually the conditions that the nursery would be, the outplanting would be taking place in those similar conditions. Is that clear? Ms. Manderson?
Yes, it is. Thank you.
Okay, Jody and Shirley, you may go ahead. Julianne? Julianne, okay.
Good afternoon. Okay, for the SBA, my topic is on deforestation, and I'm in a farm at Stolbrook Manor. I was told that for the research, I'm supposed to have a picture showing everything before vegetation, before deforestation, and I'm asking for any source that I can get a picture from before deforestation started. Miss, I was looking, but I can't find anything, so I was told to ask the NEPA department if there's any source I can go to.
Let me clarify. A photo of a particular deforested area. That's the question. That's what exactly it is that you want.
Yes, miss.
What you can do is right. So in terms of all the specific information in terms of where exactly it is or where you want this particular photo from your site, that would need to be provided. Now, there are two things. You can send your query via email to us and we will be able to say whether or not we have that. Or you can also contact the forestry department. Now, in previous webinars or previous seminars, we would have presentations on deforestation. Usually we go through either the forest department or the Department of Geography at UWI. This year we had to condense the program, so deforestation is not one of the presentations that we have now. But I do encourage you that you can reach out to either us or the forestry department, and then you will need to include the specific information in terms of where exactly and what sort of information it is that you need. So I hope that answers your question. Okay, so Mr. Downey. Okay, you can go ahead after Mr. Downey. I think we can take one other question after that, and then we move to closing. So Kenardo.
Good afternoon. This question is for Miss Watson. When she was presenting on the coral reefs, she mentioned that temperature can also affect the life of the corals. I'm asking if since we're struggling with global warming, if global warming can also be a major effect on the corals as well, as it relates to increasing the temperature of the oceans.
Good afternoon. Yes, global warming, which is considered under climate change, does have an impact. And as I mentioned, I did speak about sea surface temperature monitoring, which is something that we started doing in 2013, and that tracks the changes in the annual temperatures that we are seeing on the reefs that we have locally. So that will give you the minimum temperature, your maximum temperatures, as well as give you an average of what you see on a day-to-day basis. So the last graph in the presentation that I had shown showed where the temperatures were the highest, and all of the highest temperatures were above the 30-degree threshold, 30 Celsius threshold, which are above the coral threshold in terms of coral survival. So the longer temperatures are above that are exceeding 30 degrees, the more likely it is that there'll be coral bleaching. The higher global warming contributes to warmer sea temperatures, and that contributes to coral bleaching, which contributes to coral death. Is that clear? I'm going to assume yes.
Yes, miss.
And so we're able to accommodate one more question from Samantha. Samantha, you can go ahead.
Good afternoon. Like about the SBA, the word limits. Will we be penalized if we go over the amount with the words?
Okay, so Mrs. Russell will be responding to that question.
All right, will you be penalized? But before you consider that, I would say don't go over the word limit. All right, but you are allowed 200 words over that 1,000. So I would advise you to check as you go along, as you do each component of the SBA, you actually make a check in terms of the number of words that you have so that you're not going too much over that 200. But we do apply penalties for going too far over that 1,000 word limit. All right, so try your best as possible to keep within that 1,000 word. All right.
Thank you, Ms. Russell.
Okay, so before we wrap up, we want to hear from you. So we should be having our final poll for this session, and that should come up shortly. And we encourage you to respond. We want to be able to say, you know, for us to get feedback from you or participants, you know, what aspects of the session were useful to you, and so that we can improve the session going forward. So there is, so the poll should be coming up shortly. Okay. Okay. So, so what we're going to do now is just to wrap up. I want to thank all our presenters. I want to thank Ms. Kayan Miller in terms of going through and giving us an idea of how we can access resources from NEPA. We encourage our students and our teachers to reach out to us via email or telephone in terms of accessing resources for their presentation. I want to thank Mr. Thorney for joining us all the way from St. Anne and giving us an overview in terms of mangroves. We had Mrs. Safia Roden, who went through water quality testing and sampling. I hope you would have learned something in terms of what you need to do if you have to collect samples of your own. Miss Gabrielle J. Watson, going through coral reefs. I realized that a lot of persons had a lot of follow-up questions for her in terms of coral reef. And last but by no means least, Mrs. Russell, giving you guys guidelines in terms of what you need to do to ensure that you get maximum marks going into your exams later this year. I know it's difficult, it's a difficult time to be, you know, going through school and learning, but we appreciate the fact that you were able to join us today and that you found the session useful. By all means, reach out to us, let us know any feedback you have in terms of this session, anything we can add, anything that we can improve as we seek to provide support for you as you move on to the next stage of your education. So thank you everyone and have a wonderful day.
A coral reef. It can be one of the liveliest places on Earth. But what's really surprising is how much more goes on here than meets the eye. Even the corals themselves are alive. They're made up of tiny animals called coral polyps, distant cousins of jellyfish. Hundreds Even thousands of polyps can make up a coral. And this impressive reef, they built it, millimeter by millimeter. How? A polyp is mostly stomach, with a mouth on top. It sits in a hard skeleton that's part of the reef. The polyp takes in dissolved minerals from the water and mixes them with proteins. Then it rises up out of its skeleton, leaving space below. It deposits calcium carbonate, also known as limestone, into this space. Over time, each little polyp not only builds its own skeleton, but adds to the structure of the reef. So what gives the polyp the energy for all this building? A unique partnership. Inside most reef-building polyps are tiny algae called zooxanthellae. They use energy from the sun to photosynthesize, producing up to 95% of the food the corals need. As animals, the corals emit nutrients valuable to the algae too, and also give them a protected place in which to live. This trade-off benefits both the algae and the corals. It's called a symbiotic relationship. The algae are even responsible for the colors we identify with corals.